Happy day, I am Haripriya, Assistant Professor in the Department of Food Science. In today's class, let's discuss on the topic gluconeogenesis. In mammals, some tissues depend almost completely on glucose. For example, the human brain, erythrocytes, nervous system, testes, renal medulla and the embryonic tissues are completely dependent on glucose as their fuel. The brain requires around 120 gram of glucose per day, which is nearly half of the glycogen which is stored in the liver and in the muscle. During exercise or during fasting or prolonged starvation, the glycogen store gets depleted. In this case, our organism needs a method for synthesizing glucose from a non-carbohydrate precursor, and this is accomplished by a pathway called gluconeogenesis. Gluconeogenesis is a process of converting non-carbohydrate precursors to glucose or glycogen. The major site is the liver and the minor site is renal cortex or the epithelial cells that line the inside of the small intestine. The major substrates for gluconeogenesis is glucogenic amino acid, lactate, glycerol and propionate. These non-carbohydrate precursors of glucose are first converted to pyruvate, or they enter the pathway at the later intermediates such as oxaloacetate or dihydroxyacetone phosphate. Let us look into the precursors of gluconeogenesis in detail. First, the glucogenic amino acids. Amino acid catabolism serves as a main source of pyruvate and oxaloacetate for gluconeogenesis during fasting or during carbohydrate starvation. Amino acids that get converted to glucose are usually termed as glucogenic amino acid. Alanine and glutamine are very significant glucogenic amino acids. Now let's look into the picture. You can see alanine, cysteine, glycine, serine, threonine and tryptophan can get converted to pyruvate, and that pyruvate can become oxaloacetate and then form phosphoenol pyruvate and then enter into the gluconeogenesis to form glucose. For example, asparagine and aspartate can get converted to oxaloacetate, and oxaloacetate again can form PEP and then to form glucose. The next important precursor for gluconeogenesis is the lactic acid. Okay. After vigorous exercise in our skeletal muscle, anaerobic glycolysis happens and pyruvate gets converted to lactate, and this lactate is shuttled out into the blood and it is taken by the liver. In the liver, lactate gets converted to pyruvate and pyruvate enters the gluconeogenesis process. This cycle we usually call it as Cori cycle or sometimes as lactic acid cycle. The next important precursor for gluconeogenesis are glycerol and propionate. Glycerol is an excellent substrate for gluconeogenesis. It is generated by the hydrolysis of triglycerides. Triglycerides on breaking down forms free fatty acid and glycerol. Glycerol is shuttled into the liver. In the liver, glycerol gets converted to glycerol phosphate and glycerol phosphate becomes dihydroxyacetone phosphate. Now, the odd chain fatty acids yield either propenyl-CoA or succinyl-CoA. The propenyl-CoA is catalyzed by the enzyme propenyl-CoA carboxylase to form methyl-malonyl-CoA. Methyl-malonyl-CoA can be converted to succinyl-CoA and the succinyl-CoA enters the TCA cycle. So far we have seen the important precursors of gluconeogenesis. Now let's move on to the pathway. Gluconeogenesis and glycolysis, they share the same pathway, but they operate in opposite direction. It happens in the cytosol and they are reciprocally regulated. Out of the 10 steps, 7 steps are shared by our glycolysis and gluconeogenesis, but in the opposite direction. Okay. The 3 non-equilibrium reaction needs to be bypassed in our gluconeogenesis process, and that's the reason why gluconeogenesis is not just the reverse of glycolysis pathway. Now let's look into the flow chart. In our glycolysis, the first step, conversion of glucose to glucose 6-phosphate by the action of the enzyme hexokinase. This needs to be bypassed. The second step, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by the enzyme phosphofructokinase. It's again an irreversible step. And the last reaction, conversion of phosphoenol pyruvate to pyruvate by the action of the enzyme pyruvate kinase. These are the 3 irreversible step in glycolysis which needs to be bypassed in our gluconeogenesis process. All of you should be knowing that Breaking down glucose to pyruvate is a process of glycolysis. Now, what is gluconeogenesis? Conversion of pyruvate to glucose is a gluconeogenesis process. Now, let's look into the three reactions which we need to bypass. The first one is pyruvate needs to get converted to phosphoenol pyruvate, and this happens in two steps. The first step is pyruvate gets converted to oxaloacetate. Pyruvate carboxylase is the enzyme for it. Now, this oxaloacetate gets converted to phosphoenol pyruvate by the action of the enzyme phosphoenol pyruvate carboxykinase. So, in this two-step process, we spend around 6 ATP for this conversion. So, the first bypass reaction is done. The second bypass reaction is conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. The enzyme fructose 1,6-bisphosphatase cleaves the phosphate in the first position and we get fructose 6-phosphate. Now, the fructose 6-phosphate gets isomerized to glucose 6-phosphate, and the third bypass reaction is where glucose 6-phosphate by the action of the enzyme glucose 6-phosphatase forms glucose. So, these are the three important bypass reactions of gluconeogenesis, which makes gluconeogenesis process as an irreversible process. So, to have a short recap, the first step is pyruvate to phosphoenol pyruvate. Two enzymes are involved, pyruvate carboxylase and carboxykinase. The second enzyme is fructose 1,6-bisphosphatase, converting fructose 1,6-bisphosphate to fructose 6-phosphate, and the third reaction is glucose 6-phosphate getting converted to glucose by the action of the enzyme glucose 6-phosphatase. When we talk about gluconeogenesis, it is a very expensive process, but it is essential. The high energy cost invested in gluconeogenesis is to ensure the process is irreversible and also to ensure the supply of glucose, which is very, very important. When you look into the overall reaction of gluconeogenesis, we can see that we have invested around 6 ATP for making one glucose molecule. Coming into the regulation, glycolysis and gluconeogenesis are reciprocally regulated to prevent the wasteful operation. In well-fed state, glycolysis is on and gluconeogenesis is off. During starvation, gluconeogenesis is fully on and glycolysis is off. Insulin stimulates glycolysis and glucagon activates gluconeogenesis. Looking into the enzymatic regulation, phosphofructokinase, the very important regulatory enzyme in glycolysis, is stimulated by M.P. Fructose 1,6-bisphosphatase, the important enzyme of gluconeogenesis, is inhibited by M.P. Fructose 2,6-bisphosphate levels regulate both glycolysis and also gluconeogenesis. High levels of fructose 2,6-bisphosphate stimulates glycolysis and inhibits gluconeogenesis, and low levels of fructose 2,6-bisphosphate stimulates gluconeogenesis and inhibits glycolysis. Now, to have a short recap, we have seen gluconeogenesis as a process of converting non-carbohydrate precursors to glucose or glycogen. It is of particular importance when carbohydrate is not available in our body. The significant substrates are amino acids, lactate, propionate and glycerol. The important site of gluconeogenesis are liver and kidney. Since glycolysis and gluconeogenesis share the same pathway but in opposite direction, both are irreversible process and some of the important reactions are bypassed. Hope the concepts were clear. For more such videos, like, share and subscribe to M.O.P Classroom. Thank you.